Hi, I'm Samantha. I'm a fashion model, scuba diver, and pro volleyball player. But my favorite thing to do is study human anatomy and physiology at SEC. One day I hope to find a cure for cancer. But right now, I'd like to tell you a little bit about skeletal muscles, how skeletal muscles contract, and the molecules essential to the process. Here we see the axon of a nerve cell that has received a signal. This signal is the action potential that travels down the axon, opening up calcium channels along the way. As calcium ions flood into the axon, the synaptic vesicles containing a neurotransmitter called acetylcholine get pushed down toward the synaptic knob. These vesicles fuse with the axon's membrane, releasing acetylcholine into the synaptic cleft. This chemical signal travels across the gap between the synaptic knob and the motor end plate in the sarcolemma of a muscle fiber. As it contacts the motor end plate, it hooks up with receptors and causes them to open. Sodium ions present in the cleft now flow freely into the muscle cell, causing a big action potential. Let's step over and get a three-dimensional view of the process. Here we have a clay model of a whole muscle. The whole muscle is covered by epimysium. This is the connective tissue that continues on beyond the muscle and connects to a bone, called a tendon. Inside we find many bundles of muscle cells called fascicles. Fascicles are covered by perimysium, seen here in orange. Inside the fascicles we find individual muscle fibers, each covered by endomysium, seen here as green. To find out how the action potential causes contraction, we need to zoom in a little more to get a close look at a single muscle cell.
Okay. Was that good?
Yeah.
It's still recording.
Oh.
At our next clay model, we can see that under the endomysium is yet another layer of membrane called the sarcolemma. Here is where the axon delivers its signal to the muscle. The action potential travels down the sarcolemma and into the T-tubules, which are invaginations of the membrane. On either side of a T-tubule is a modified endoplasmic reticulum called the sarcoplasmic reticulum. Massive amounts of calcium is stored here in the terminal cisternae of the sarcoplasmic reticulum. This calcium is being held in by a molecule called calsequestrin. The action potential causes the release of all this calcium into the sarcoplasm of the muscle cell.
It might help you reach up.
For our next segment, we need to move to our special pipe cleaner model. Here we find the thick and thin filaments of an individual muscle fiber represented by colorful pipe cleaners and beads. The ATP produced by the mitochondria within the cell energizes the myosin heads as ATP is converted to ADP, putting them in a high-energy state ready to connect with the actin. Before they can connect, however, we need the calcium that has been released from the sarcoplasmic reticulum to attach to the troponin complex, causing the tropomyosin to move and reveal the active site on the globular actin. The myosin heads attach to the actin, forming a cross bridge. As the myosin pivots back to the low energy position, they release the ADP and move the thin filaments to overlap the thick. This is called the power stroke. This pen and paper illustration shows how the thin actin filaments and Z discs move toward the center M line, and where the A band, I band, and H zones are located in the sarcomere. After a muscle contracts, another ATP is required to detach the myosin heads from the active site of the actin, breaking the cross bridge. Now the myosin head becomes energized again as ATP is converted to ADP. Calcium is then pumped back into the sarcoplasmic reticulum, allowing the tropomyosin to again cover the active site of the actin, and then the muscle relaxes. Acetylcholinesterase in the synaptic cleft destroys the acetylcholine and ends the signal. We'll see you next week when we'll be looking at the medulla oblongata of the sheep brain.